Thank you, Ryan. Thanks for inviting me. Actually, it's mainly going to be about SpikeInterface. I have a couple of slides on SpikeForest as well, but mainly SpikeInterface. And so this is going to first introduce SpikeInterface, how the project started, and what we try to solve. And in the second part, I'm going to show you some more recent advancements that we are doing in order to make SpikeInterface more standardized tools for electrophysiology analysis. So it all starts from here, and I guess from yesterday, you realize that this is not a trivial problem, right? So we have extracellular electrophysiology data. Now with recent neuro technologies, we can record neurons at unprecedented yield, right? And an essential step in processing, virtually for all experiments, is extracting single neuron activity from these like extracellular traces that contain a mixture of activity from multiple neurons. When I first started my PhD in 2016, we had to choose like which spike sorting pipeline to use, and I also tried to develop some spike sorting analysis techniques, and I soon faced this problem. There was a jungle of tools out there, really a plethora. I mean, now probably things have gotten a little bit better. Many of these tools maybe are not as maintained as before, but my first reaction was getting a bit lost. I guess maybe you share this experience with me as well. And this because it's very hard to actually address electrophysiology data. There are some inherent complexities and challenges in the field. First of all, and maybe differently than imaging, we have really many different acquisition systems which come with their own different file formats, right? So you need like readers to be able to interface with different acquisition tools and so on. And building software that's flexible and can accommodate for all these different formats, it's a difficult task. The second thing is on the spike sorting side. So many of these tools, they are actually built in different technologies. They may rely on C and C++ for speed. There may be MATLAB-based, so you need a license to run them. Or they're built in Python, but still there's a large variety in the implementation and also in the configurations files and all the parameters that you need to provide to the sorter, what kind of file format the sorter expects, and so on. And the first two challenges also basically bring the third challenge, which is there was a big lack of data provenance and reproducibility, right? So some papers cite that spike sorting was done the usual way, right? So we tried basically to address some of these issues. This started in 2018. It was a nice collaboration with many different institutions. And we built SpikeInterface, which is basically a Python package to run multiple sorters on the same data set with very minimal effort. On top of this, we took the chance to actually also bundle additional functionalities that are needed in the spike sorting pipeline, which is not spike sorting per se, right? You might need to pre-process your data, to do some denoising, to extract features and other additional data after spike sorting. You might want to do some spike sorting comparison. Say you run a spike sorter with two different sets of parameters or two different spike sorters. How do they compare? You can also do some data visualization on the recording, the sorting output, waveforms, and so on. And importantly, we try to make everything as reproducible as possible, and I'll touch upon this later more. So the way that we try to approach the problem is basically to try to minimize this part on the top. So as it used to be, you have your acquisition system, you write your custom code to interface from your system to a specific spike sorter, and then you have some other custom code to interface with the sorted data and continue with the downstream analysis. This makes the field very fragmented and error-prone, right? We are not all software engineers, right? So what we try to do is basically abstract away the way that we think about files, right? So now, and we built objects that basically allow you to read from over 25 recording file formats and over 20 sorting file formats in a standardized way. So you can't forget about where the data came from if you can interface with them the same way. And since this interface is standardized, we can also wrap several different spike sorters because we know the inputs and outputs. So now SpikeInterface supports over 15 spike sorters that you can really run with a single line of code. And in addition, we also try to build standardized, tested, and community-validated tools, as I said before, for pre-processing, post-processing, quality metrics, automatic curation, and visualization. And I want to stress this, really the community effort, because we can all, especially with these kind of like events as well, we can all come together and try to basically make things better and easier for new labs, for new PhD students, because we face these challenges. We know it's been hard, not only in extracellular electrophysiology, but in general. Just to show you an example, so this is what like a PhD student might want to do. They perform a recording, say using the OpenEFIS acquisition system. They want to read the recording first, maybe input some probe information. Maybe they're using tetrads, so you want to tell the sorter you have like different groups made of 4 electrodes. You might want to filter your data, remove some noise, common median reference or other denoising, removing artifacts, and what's more, running mountain sort, for example, extracting the waveform snippets, from them computing some quality metrics, and exporting everything to Phi, which is a very common and widely used software for spec sorting, visualization, and manual curation. So these 8 lines pipeline, 8-step pipelines literally translates to 8 lines of code. So it's a very high-level API, and it's very user-friendly. So it's really thought for like biologists or neuroscientists which weren't trained to like in coding. So it makes everything very declarative. So you read OpenEFIS, set the probe, bandpass filter, common reference, and so forth and so on. In addition, since now we have this tool to be able to run multiple spike sorters on the same data sets, we started looking into spike sorting comparisons at scale. And this was a collaboration also with the Flatiron Institute and Jeremy. Basically, in this spike sorting jungle, can we pick up the lion? What sorter should people use? And first of all, we observed that spike sorters strongly disagree. So here we basically ran this 6 different spike sorters on the same data set. It was a NeurPixels data set from the Allen Brain Observatory. And we ran 6 sorters, herding spikes, kilosort, iron class. Actually, this is kilosort 2, iron class, 3D clue, spike and circus, and HD sort. And here in color code is the number of units that K sorters agree upon, right? So overall, over like 1,500 units were found by pulling together all the units from different sorters, out of which only 33 were in agreement with all 6 sorters, right? This might also mean that some of these sorters are bad, right? It's not, so maybe spec sorting is bad, you know, but now it's fast. But still, this was like a very staggering result. So then we kept collaborating with the Flatiron Institute, and they actually led this effort. It's called SpikeForest, and basically we gathered over 1.3 terabytes of ground truth data sets, so either paired recordings with like ground truth patch clamp or juxtacellular recordings, or simulated data, some hand-curated data. And then we basically ran all the available spike sorters, basically, and they, especially Jeremy here, built a very nice interactive website where people can actually explore the results and maybe pick the best sorter for their experiment. Say they're using tetrads or NeurPixels, you might want to choose a different spike sorter. And this also motivated a big push in spike sorting development, because if you see a light blue here and you are the developer of 3D clue, you go back and you try to improve it, right? So this is really pushing the field forward. So in the second part of this talk, I want to talk about some recent features that we added to SpikeInterface to make it basically cloud-friendly, and because the Allen Institute of Neurodynamics, for neurodynamics, we're basically moving all the processing to the cloud. This is what we are doing. So basically, our scientists, they sit at the rig, they do experiments. Then the first thing we do, we transfer the data to the cloud. And this triggers automatically a computational pipeline that is run on this CodeOcean platform. And you've heard yesterday from David a little bit more about it. And also the final part, so the downstream analysis is basically interacting with the cloud through standardized data formats. So the first thing that we tried to solve is basically to minimize the data footprint of electrophysiology. So we looked at data compression, and we implemented in SpikeInterface a compression framework that's based on ZAR, and we tested many different possible compression strategies. It turns out that if you use audio compression for electrophysiology, it works great. So we extended ZAR by including some custom codecs, including this WAFPAC audio codec, which performed best among all the tried options. And we can compress now NeuroPixels 1.0 data down to 27% in file size, and for NeuroPixels 2.0 down to 40% in file size, due to some inherent differences in the two probes. And if you want to know more, we recently published a paper with each as a benchmark. And I also want to highlight, also in this case for compression, it's very easy to run. So you just read your recording, you define any ZAR-compatible codec, and then you save to ZAR, applying a compression. You can also use parallel writing to do this. Another thing that we are trying to do, and I think we are in a key position to do this, being a new institute that basically plans to acquire and analyze large-scale recordings, is trying to build standardized and efficient pipelines for the community. And this would basically lower the barrier for adoption and also increase the possibility to compare data that is analyzed across different labs. In addition, having a centralized resource for this kind of like very basic, it's basically pre-processing, doing spike sort, and then you do the fun stuff later, right? It would also facilitate the deployment of new tools. Say now Kilosort 4 comes out or some other tools, we can easily push an update, we can push a new drift correction method that performs better than what's out there, and so on. In addition, everything can be version controlled, right? We can have both code and dependencies, we can have Docker containers and more, and overall this would mean better provenance and reproducibility in the field. So we started this is still a work in progress, but we have some working repos that we're currently using, and we are now in the process of extending it and making it more flexible for people to use it. But this is how it looks. You can either run this script, this pipeline as a Python script or as a Nextflow workflow. So in the latter case, each of these steps, like pre-processing, spike sorting, post-processing, curation, and visualization, can actually run on different machines with appropriate computational resources. For example, a GPU is really only used by the spike sorter. So why paying for a GPU for pre-processing if it's highly CPU heavy? One problem that we faced and we tried to solve, again, in collaboration with the Flatiron Institute and Jeremy, is this visualization. So if everything runs in the cloud, how do we provide powerful and interactive visualization? So we collaborated with Jeremy and basically co-developed this. Jeremy developed the technology. We helped in like designing these views, but we're using this Figurl technology, which basically uploads the plotting data to the cloud, and then when you get one of these, it makes a shareable link, and then a web application can read the data and renders the plotting data with appropriate visualization plugins. And we're using these for quality control and manual curation. And also I can just send you some of these links and you can check them out, but I want to show you real quick how they look. So we use them both for like time series quality control. This, for example, is a NeuroPixels recording, and we have for two different half-a-second segments. Basically, this is the raw data, and you can also show different layers. So it's the raw data, phase shift, and high-pass filter data. And this on the right is basically pre-processed data. So we go from this high-pass after removing the channels that are outside of the brain. You see, you don't have this like flat band on the top, and you can see how CMR cleans up the signal, or a high-pass spatial filter cleans up the signal. And we can also visualize like drift maps to see how the data is changing over time. On the spike sorting side, we tried to make a web-based version of Phi. So here, basically, you have here a list of units that you can scroll through. So this is unit 0, unit 1, 2, 3, and so on. You can see their waveforms here, their templates, their distribution here, auto and cross correlograms as well. If you select more than one unit, for example, for this unit 4, it seems to be incomplete. So you can sort here by similarity. This unit 8 seems to have a very high similarity score. Oh, they're probably a merge, right? And you can also do manual curation. So in this case, we'll say, let's merge, and let's accept the new unit as a good unit, and then we can basically export this curation to push these curation files. They're just JSON files to a GitHub repo that we use to store all this curation data. And internally, that automatically triggers an action that re-uploads the data to the cloud for the scientists to use it. Okay. So coming back here, I had a GIF here in case things didn't work. They worked. Again, what we're doing now is mainly using CodeOcean as a backend, but we are actually, this is still work in progress, but we want to make sure that these standardized pipelines can also be run with different backends, like using local resources if you have invested in a very powerful machine in your lab, using Slurm if your institution invested in a very powerful computational resource, or directly using some cloud solutions like AWS, Azure, or Google, or whatever is available. So to recap, I hope I showed you that SpikeInterface is like now a mature ecosystem for electrophysiology data analysis. It's heavily community developed. It's user-friendly with its high-level API, making basically approaching this complex analysis very easy. You can access multiple sorters and the latest state-of-the-art processing tools, and it also enables large-scale comparison of these tools. And in the second part, I wanted to show you some recent additional features that we developed to make SpikeInterface cloud-ready. So compression to minimize the data footprint in the cloud, efficient, parallelized, and reproducible pipelines, and these web-based shareable visualizations. Overall, we hope that with all the community basically collaborating on this, that we could provide standardized pipelines for electrophysiology data analysis. Finally, I want to thank the many users and contributors, especially Samuel Garcia. Here is the main other core developer of SpikeInterface, all the funding that is helping us to grow, and thank you all for the attention.
We have time for 2 questions for Alessio.
How do you store the metadata here? Let's say I run this pipeline and I do some analysis, and then now I want to run different spike sorting algorithms to the CodeOcean. How do I get access to that?
So there's 2 levels of metadata. So one level is, let's say, the session level subject, and I think David covered that yesterday. So we're building these data schemas for all kind of experiments. And then basically, what this is doing is still using the data schema that we developed, but adding like processing steps. So each of the steps that I showed before in the pipeline will basically add a data process to a processing schema, with basically spike sorting parameters, spike sorting logs, all the parameters used for pre-processing, post-processing, and so on. And this could also be further developed to be like, let's say, standardized schema that then could also be rendered as web forms if you want to change them. And so one could provide like a standardized set of parameters, and you want to change the frequency that you want to filter your data with, you should be able to do it. Right now, we are still working on it, but yeah, that's the idea. But that, I think, where we need community feedback. So what parameters should we expose, and like what should we?
I think everything, because you go through a very expensive pipeline. This is my experience. You want to maintain the integrity and have access to all the metadata.
So, yeah.
It's too expensive to go back.
Yeah. So maybe everything, but hidden by default. Yeah, no, sure, in terms of that.
So it's just a matter of organizing.
Yeah, I would say that for an end user, it might be a bit overwhelming to see this, like, I mean, there's lots of metadata and parameters going through this, right? So maybe you start with, if you know what you're doing, and it takes time and experience to know what you're doing, I think you should have the possibility to change things. I agree. Yeah, Mackenzie.
Yeah, thanks for the really cool talk. This is really inspiring work, so thanks for doing it.
Thank you.
It's probably not always as thanked as it should be. Comparisons are awesome.
Thank you.
So I actually had a kind of maybe a technical question about this comparison of all the different spike sorters where you get down to the 33.
Yeah.
So I was sort of struck by it's a very short data set, 15 minutes.
Yeah.
And so I'm just wondering if you had more time, like, right, in some sense, like as Andreas mentioned, we're always trapped in space and time. And record. So does this hold true if you had, I don't know, 6 hours of continuous?
I think it would be worse. Yeah.
Okay, so that's what I wanted to know. So you think that if anything, we should be spike sorting in much smaller bins and then trying
No, but then you would have so it would be worse because you probably would have like some drift, or so then you'd need some appropriate drift correction methods. But then, so 15 minutes, and this data set, it was actually handpicked because it was quite stable. But if you had to do the other way around, this actually, for some people that do, for example, 48 hours neuropixels recording, this question comes up: how should I approach this? Like, is it like one record, or should I split this up and then try to like connect the dots? The other problem is that if you split it up, then you're facing the problem of how you connect the units found in the different sessions. I mean, you could do some overlap and try to do some matching and so on. It's still, I would say, very ongoing and under development.
Maybe that could help with this problem. So I was just, but I'm happy to chat.
Could be interesting.
Yeah.
All right. Thank you.